I'm Uta Francke. I'm a human geneticist, originally from Germany, as you can hear. And I'm an emeritus professor here at Stanford and also senior medical director at 23andMe.
Tell us when you first fell in love with genetics and when you started studying and sort of your journey to where you are today as a Stanford professor.
I went to medical school in Germany. And then I trained in pediatrics. I came to this country to train at Los Angeles Children's Hospital. And then I had to think about what else to do. And what I really wanted to do was endocrinology, because I loved biochemistry. I thought biochemistry was the best thing in medical school. But I couldn't get in as a foreign medical graduate. And genetics was just in the beginning, started to get interesting. It was at the time where it became possible to identify chromosomes by chromosome banding. And there weren't many people going into that field, which had a great advantage, because as a fellow at UCLA in human genetics, I was allowed to do anything. I could learn chromosomes, biochemical genetics, as well as see the patients on the wards where people said, oh, what is this malformation? Is it a syndrome? Is it genetic? Will it happen again? So the training involved from the beginning clinical genetics and the other subspecialties of genetics.
Were you able to interact in I guess, medical school and beyond as you were sort of discovering the area that you were sort of going to focus on for genetics? At the same time, were you able to experience the clinical side of genetics as well?
All the time. I was active in clinical genetics for the last 30-something years.
And what does that involve exactly?
Well, in pediatrics, you're mostly focused on children having problems. So either children who inherit something, or children who get very sick after they are born because they cannot metabolize protein, there's something wrong in their metabolism, or children who have certain birth defects. Children who don't do well in school, who are late walking, who are late acquiring skills, or have neurological symptoms, seizures. So all kinds of problems that people didn't know what is this. So there was a lot of discovery to do. We found out that certain clinical phenotypes were associated with chromosomal duplications or deletions, now that we could look at chromosomes in higher resolution. So that was the first stage, so in the 70s, make discoveries this way. And now, of course, whole genome sequencing is applied to these children. And when nobody could figure out what caused their problem, you look at their whole sequence and you can find a change. And that's been very successful in the last couple of years, since high throughput sequencing methods were developed. So the exciting thing about my own career is the two-way street. It's always been two ways between the clinic and the laboratory. You see somebody fascinating in the clinic and you bring samples to the lab and you tell your PhDs, look, this is what the patients look like, and we have to figure out what's going on here, and that gets everybody motivated. When you learn something in the lab, you find a gene, like we found a gene for Rett syndrome, and then you can figure out, can we do anything to help these patients? Now that we have the gene, we figure out its function. In the last few years, we spent a lot of time making mouse models, because we looked at diseases that involve the central nervous system, and you can't go into a human brain and take a sample. I mean, some people have part of the brain removed because of some medical indication, and then of course you can get it. But what we did, make mouse models, because the genome is very conserved between the human and the mouse. And even though we're working with deletion syndromes where part of a chromosome is missing, we can define where in the mouse genome that part is located. It's got the same genes in it, take it out in the mouse, and then you have mice that you can study their development prenatally, afterwards, take the brain, cut it up, look at what's going on. And this is what we did mostly in this millennium, I would say.
What would you say is your general research subject?
There were too many to mention. I really started out with whatever was possible to do at the time. You know, I was in a way driven by technology that was being developed, and I saw where this technology could be applied to the problem. So it started with cytogenetics in the area of banding chromosomes. That was in the 70s. And then biochemical genetics, somatic cell genetics, meaning that you can fuse cells from different organisms and you can map chromosomes. So in the beginning of the Genome project, we were very much invested in mapping genes to chromosomal regions, with the idea, if you have a gene that's been cloned and you know the function, you map it to a chromosome region where we already know a certain disease gene has been mapped, then becomes a candidate gene for this disease. And that was a useful approach in the early 90s. And then as all these other technologies became available to study global gene expression patterns and doing mouse modeling, and then we moved to studying individual diseases, like Rett syndrome and Robert syndrome, is a malformation syndrome. And ultimately, I thought the deletion syndromes, where you take out a chunk of chromosome, and would be interesting because there are many different genes in here. And which of these genes contribute to what part of the phenotype? I found that fascinating. And that's was the projects we're working on in the end. So it's a stepwise process of discovery that makes use of the technology as it is being developed.
So thinking about what you do as a career, what do you think the future is for people who are interested in being clinicians and bridging the gap between people and the research that can help them? Do you think there's going to be a bigger opening in the future for people who want to do something similar to what you do?
Well, there is definitely a need, and there are many opportunities. But the drawbacks are in genetics, for example. We don't have enough people coming into medical genetics out of the medicine side, because in genetics you don't make any money. We don't have procedures. We don't stick tubes into people that we can charge for. It's all cognitive. You spend a lot of time going through complicated information, searching the literature, sitting down with the family, counseling them. You don't get reimbursed much for this.
Sure.
And therefore, it's not attractive, not for people who went to American medical schools and to enter a career as a medical geneticist. So we have a shortage, and many of the most successful people are foreign-trained medical doctors who don't have to pay for their medical education.
Well, we hopefully have a very large international audience, participating in our course. What would you say to a student or a lifetime learner who's watching and is really interested in the subject and may even be interested in thinking about this as a career? What advice would you give them?
I mean, I can only say this has been a lifetime learning experience to be part of genetics during those decades when everything developed, and we are not done yet. I think it goes on and will be even more exciting. There are many secrets in the genome that are yet to be unraveled. And both PhDs and MDs should consider this. And then genetic counseling, that's a whole new career path. We have a training program here at Stanford for genetic counselors. It's a master's program. Because you don't have to have a PhD or an MD to tell people, this is what we found in your sequence and this is what it means. And these people are trained specifically to do that, and we'll need a ton of them, lots of them. So I want to encourage people to consider this as a career.